Hello, this is talk number 33 in the series from Principles of Biochemistry, written by Albert Lehninger, and the talk is coming from the 7th edition. Today we'll be discussing RNA processing and elements of translation, the production of protein from a messenger RNA template. And RNA is an interesting molecule, much in the news. It has multiple functions. One, RNA can act as a catalytic enzyme, which we call a ribozyme. The primary transcript is form of newly formed RNA, and the RNA can be spliced to produce a final transcript. The 5' end has a protective 5' cap, which is the 7-methylguanosine, which protects against ribonucleases. This mechanism is utilized by influenza virus to maintain viability. The RNA transcribes introns, which are non-coding, and exon-coding sequences, and the intron sequence is spliced out. These have a regulatory function. The complexes, we call these spliceosomes. Messenger RNAs, such as in eukaryotes, have a polyadenine 3' tail, and this binds the proteins and protects from nucleases. Alternative splicing is where a gene can give rise to multiple products based on the sites of the splicing. And processing also affects ribosomal RNA. These originate as complex riboproteins which undergo transformation. So we talk about the structure here. We have, for example, a 90S pre-ribosome. This is the method of having post-transcriptional modification. There are various components to the RNA gene. You have methylation, pseudouridine formation, and other modifications. These convey permanence to the RNA template, so it's not digested by ribonucleases. And this requires, a series of ribonucleotides. You get pre-cleavage, initial cleavages of pre-RNA with addition of other protein moieties here. You have pre-40S, pre-60S, and additional cleavages occur with nuclease processing, give you finally the 60S and 40S ribosomal subunits. These are complexes of protein and RNA, and these are active in translation of protein synthesis. Transfer RNA occurs as a longer sequence prior to processing. The RNA enzymes, the ribozymes, are engaged in splicing introns, RNase P, and a hammerhead ribozyme, which assists viruses in replication. It is shaped like a hammerhead, and these are small RNA sequences that we call virusoids. Messenger RNA degradation. Messenger RNA has a very short half-life and degraded by ribonucleases. The synthetic RNA polymers are catalyzed by polynucleated phosphorylases. And the central dogma that had ruled biochemistry for decades was that our DNA was transcribed to RNA, translated to protein, but the discovery of reverse transcription, where RNA can be reverse transcribed to our DNA, turned that central dogma on its head. So when we look at DNA replication, we can produce more DNA. We can transcribe DNA to RNA, messenger RNA specifically. Reverse transcription gets RNA back to DNA. This is seen in viruses. In many of them, their RNA viruses function through the DNA reverse transcription. Also, RNA can be in the transfer RNA form can be utilized along with messenger RNA template to produce protein, and you can also have RNA replication. So this allows viral RNA to replicate. They're retroviruses, for example, human immunodeficiency virus. This is an RNA virus which contains reverse transcriptase. There are 3 genes that catalyze RNA-dependent DNA synthesis, RNA degradation, and DNA-dependent DNA synthesis. So this is an example of a retrovirus. This is a transfer RNA here. This is the RNA genome there. There's reverse transcriptase, integrase. There are proteases with this. There is a GAG protein envelope receptors, and this is an example of a retrovirus. So there are receptors that interact with the plasma membrane, and reverse transcriptase creates a viral RNA from the messenger RNA or the RNA template. This integrates into the host genome, is transcribed, and this virus will be transported out of the nucleus, encounter a ribosome. You get translation, proteolysis, along with the messenger RNA, which can be produced in large quantities and break out and destroy the nucleus. And this is assembled, packaged, and released back into circulation where it can spread to other sites. And if it is a communicable virus, it can be spread to other host systems. Telomeres are bases at the end of the chromatin strand in meiosis. We'd seen these previously. They require an RNA primer to create additional DNA. This is an analog of reverse transcriptase. Telomerase is the enzyme involved. The telomere length confers organism longevity, and with age, these telomeres become shorter and more subject to mutational errors. RNA-dependent RNA polymerase is produced by some prokaryotes and viruses. RNA occupies a central role in transcription, translation, catalytic activity, and is the parent molecule in reverse transcriptase action. Protein metabolism. Proteins are the end products of information pathways. They require 90% of a cell's energy for biosynthesis. The RNA template is effectively an RNA enzyme. There were 3 advances to ascertain the genetic code. First of these was early amino acid labeling in concert with RNA centrifuge fractions. The discovery of transfer RNA and amino acyl transfer RNAs was critical to this process, and that there was a hypothesis that an RNA template coded for a corresponding transfer RNA. So there's a group of 3 nucleotides, as I'd shown earlier. There's a nucleotide triplet, and these are the codes. So you get CUA, or in this case you have ACU, before that AUG, CU, and then whatever occurred before that, AGU, UGG, and so forth. There is an adapter. This adapter is transfer RNA, and the amino acid is characteristic of the adapter sequence. So the adapter sequence triplet is homologous with hydrogen-base pairing to the nucleotide triplet sequence. So you would have a uracil here, you would have a guanine here, and you would have an adenine here. So this is the anticodon here. These are codons along here. The messenger RNA has codons. Transfer RNA has anticodons. And when you, so if you have 20 amino, or if you have 3 amino, if you have 4 nucleotides that comprise a codon, and the codon consists of 3, a group of 3, 4 raised to the 3rd power gives you 64 combinations. And these are the codons on RNA. The first letter of the codon is U, C, A, or G. The second letter is U, C, A, or G. And the 3rd letter is shown in these breakdowns. So in the first box you have UU, second box UC, 3rd box UA, 4th box UG, and then these are the 3rd letter nucleotide of the codon is here, and each of these codes either for amino acids. Some cases they're duplicates because there are only 20 amino acids. In other cases, you have an initiator sequence. For example, methionine is the initiator sequence uniformly with an AUG, and there are 3 stop codons, UAA, UAG, UGA. So when the complex encounters one of these codes here, one of these codon sequences, protein synthesis stops. So this is a codon, 3 nucleotide base sequences which code for a specific amino acyl tRNA. The reading frame is a sequential grouping of codons. They are non-overlapping. They occur in sequence. And the elucidation of the genetic code by Hargob and Khorana relied first on nucleotide polymers, then chemical RNA synthetic methods to find the template. Initiation code is AUG. There are 3 stop codons, UAA, UAG, UGA. The open reading frame is one where you have a long messenger RNA template sequence without a stop codon. So a lot of viruses, including SARS-CoV-2, has several open reading frames which code for one protein without stopping. Degeneracy is needed because you have 64 codes. 4 of these are taken up as a stop codon and the initiation code. So that leaves you with 56 among 20 amino acids. So to give an example, there's 1 each for methionine and tryptophan, 3 for isoleucine, and 6 for arginine. The genetic code is universal between species. Every species has this genetic code. And the 3rd base is redundant for its amino acyl tRNA, which implies, evolutionarily speaking, loose binding of that 3rd one to affect amino acid transfer. The anticodon, again, is this 3-base sequence in transfer RNA, which base pairs to the codon. So in the flat planar structure of transfer RNA, this is a 3' here, and the amino acid attaches to this point. This is actually a globular arrangement with 2 broad folding overs here. It's not a linear plane, but it is represented thusly. This is a sequence of anywhere from 75 to 90 nucleotides. So here's the codon here. Anticodon is complementarily base paired, and this base pair here governs this and governs what is attached here, and you get these in sequence. This genetic code is resistant to mutation. In this sense, a mutation is one where one base pair replaces another. Silent mutations, the single base is replaced, but the codon codes for the same amino acid. This accounts for some of the redundancy that's shown up by the 3rd nucleotide in the codon. Transition mutations, the purine is replaced by pyrimidine, or vice versa, and this is the most frequent type. So AG is replaced by CU in this situation, or vice versa. Frame shift mutations, where the codon sequence is shifted by 1 or more bases, and it's deleterious because the proteins are completely different, and you do not get any productive activity. If you have premature generation of a stop codon, that additionally would stop it as a frame shift mutation. The steps of protein synthesis are activation, initiation, elongation, termination, and ribosome recycling, and then folding and post-translational processing. So this is an example of it right here. So we can begin with the activation of amino acids. So this is a transfer RNA, and it is amino acylated with the amino acid that is commensurate with the anticodon sequence. This is a very conserved, virtually error-free process. The messenger RNA in the 2nd step is initiation, so messenger RNA shown here, and the amino acylated tRNA bind to a small ribosomal subunit, and then the large subunit binds. So you have an enveloping situation. This is an energy efficient process. The elongation, the successive movement along this protein complex along this messenger RNA strand is occurring. This is relatively stationary. These protein moieties move along here. As a new codon occurs, another transfer RNA with its amino acyl linkage occurs, and you get peptide bond formation occurring until the ribosome reaches a stop codon. So what happens is that this the amino acid literally binds to the next one. A peptide bond is formed, and then the transfer RNA is released. You get enough of these, you get elongation of this process, and then termination where a stop codon is encountered, and then the messenger RNA protein dissociate. The ribosomal subunits are recycled here and here, and the protein folds. It actually folds while it's in production, but the final form, and then you have post-translational processing where you can get chemical modification that's occurring, or any other process that is applicable to the situation. Ribosomes contain ribosomal proteins. These are R proteins, at least one large RNA. There are 30S and 50S sedimentation subunits. This is a functional ribozyme, as these are structural elements separate from the active site. Transfer RNA has anywhere from 73 to 93 nucleotides and is a three-dimensional structure. So here's the amino acid arm here. There's a pseudouridine C arm here. There's a D arm here, and this is an anticodon arm with a wobble position. So this allows for some sort of flexibility and facilitates binding more critically at the first two parts of the anticodon, a little bit less so at the 3' position. Some of these are post-transcriptional modifications, so you'll get pseudouridine, you'll have methylation that's occurring here. There's also an extra arm which is variable in size. This is how this looks like on the 3-dimension, where these actually fold on each other. You can see that this arm here and the D arm are in proximity to each other. So you get this sort of L shape with the amino acid here and the anticodon over here. The stages of a transfer RNA amino acid transfer: the amino acyl tRNA synthetase attaches the correct amino acid to tRNA. The ATP is hydrolyzed, AMP and pyrophosphate. There are two classes encompassing amino acids. There's a two-step proofreading function to ensure that the correct amino acid is attached. The enzyme also selects for the correct tRNA using about 10 nucleotides in the tRNA, predominantly in the amino acid arm. The amino acid is added sequentially to the C-terminus. The AUG initiation puts an N-terminus methionine residue, and the second codon for methionine codes for any internal methionine residue placements. The initial is methionine, and methionine can occur at any point after that in the growing protein strand. In the initiation step, there's a 30S ribosomal subunit, messenger RNA initiating methionine, tRNA, 3 initiation factors, GTP, 50S ribosomal subunit, and magnesium. So it looks like this, where you get IF3 and IF1 attaching to 30S subunit. Then the messenger RNA binds. This is the process here. You get GTP binding the 30S subunit, and it recruits the methionine initiation RNA to this. You get this sort of complex occurring here, and then the 50S subunit comes in with liberation of GDP and phosphate, release of the initiation factors 1, 2, and 3. And then you finally have this globoid unit that I'd shown in schematic form earlier. This is the codon. The next codon is in this sequence here, and the methionine represents the initial. The elongation requires the initiation complex. Amino-acyl tRNAs, the elongation factors have 4 steps: a binding of incoming amino-acyl RNA, peptide bond formation catalyzed by 23S ribosome, and translocation and shift to an adjacent codon. So it looks like here, and you get this is the original methionine. This is the reaction that occurs. This is the second amino acid in the sequence, and the process after it's completed with the transfer RNAs here is that the methionine, the initiation occurs. So this in the growing amino acid, the next one that occurs, this will be placed on the following one, and this methionine will be further out. So it grows from the amino terminus, and the attachments are occurring progressively from the carboxy end. And over time, you get a long peptide bond strand. There's a proofreading function with the incorrect amino-acyl tRNA dissociated prior to amino acid transfer. And if one of the termination codons is encountered, you have release of factors involved. Ribosomes are dissociated, and there is dissociation of the other translocation components. 4 NTPs per added amino acid residue are required, and you can couple transcription and translation. So to do that, you have the DNA. The DNA can produce the RNA template, and polysomes can act almost immediately on that and get increased protein. So the DNA with RNA polymerase messenger RNA can immediately be acted on with increased synthesis of the protein strand. You have protein folding and post-translational modification. The amino terminus methionine is removed. You get chemical modifications, group additions, transfers, prosthetic groups, mineral cofactors, lipid, carbohydrate moieties, all added to get the finished product. There's ribosome profiling, where the ribosome complex and associated messenger RNA are isolated. You can remove the non-translated messenger RNA with ribonucleases, and then the intact messenger RNA is the translated component. You can have numerous antibiotics and toxins act on the translation. Tetracycline blocks binding of amino-acyl tRNAs. Streptomycin can cause misreading of the genetic code. The post-translational state of proteins, a signal sequence of 13 to 36 amino acids in a protein will direct the protein to its appropriate locations. Post-translational modification occurs in the endoplasmic reticulum. There's a signal recognition particle that can interrupt protein synthesis and allow for modification, and then resume translation. So this is a case where you get the signal sequence occurring with in the cytosol, the endoplasmic reticulum then takes the growing protein and modifies it over time. And this is the messenger RNA component right here with the 5' cap, and the protein grows over time and then goes into the endoplasmic reticulum and gets revised there. You have a ribosome cycle, an SRP cycle, and the lumen. Glycoproteins are also produced in the endoplasmic reticulum. Transmembrane transport has nuclear pores, which allow for macromolecule transport across the nuclear membrane. There's protein-mediated endocytosis. Protein degradation, the half-life of a protein can vary from 30 seconds to many days. Erythrocytes have a 110-day lifespan. Proteolytic enzymes target particular proteins. Ubiquitin is a eukaryotic protein which covalently links to proteins earmarked for destruction. the 26S proteases destroy the product. And that's it for this talk. Thank you.